Hello everyone, my name is Aravind Gopi and you are watching AG Biology. Today in this video, we discuss about classic experiments in biology dealing with genetic material and its biochemical identity. The information that reside in genes when passed to new generation influences form and characteristics of individual is referred to as genetic material. Today we know that the genetic material in prokaryotes and eukaryotes is DNA, while in some viruses RNA serves as genetic material. Until 1944, it was not clear that what chemical component of the chromosomes make up genes and constitute genetic material. Because chromosomes were known to have both nucleic acid and protein component, both were candidates. Surprisingly, since proteins were known to have both diverse and abundant in cells, and much more was known about protein than about the nucleic acid chemistry, many geneticists favored proteins. In 1927, Frederick Griffith performed several experiments that laid foundation about the nature of genetic material. He was conducting experiments with several different strains of bacterium Streptococcus pneumoniae. Among those, some were virulent or infectious that caused pneumonia in mice and human. Others were avirulent or non-infectious strains which doesn't cause any illness. Actually, the difference in virulence depends upon the presence or absence of a polysaccharide capsules. A virulent strain doesn't have this capsule, or in other words, they are non-encapsulated and are destroyed by phagocytic cells of host immune system. Virulent strains, on the other hand, possess this capsule and they are encapsulated. So they escape the host immune response, thereby causing pneumonia and finally death. How do we distinguish between the presence or absence of a capsule? There is visible difference in colony morphology of virulent and avirulent strain when grown on solid agar plate. Encapsulated virulent strain form smooth and shiny colonies or S colonies, whereas non-encapsulated avirulent strain produce rough colonies or R colonies. Griffith used avirulent R strains and virulent S strains in the critical experiments. More specifically, 2 R strains and 3 S strains respectively. If you inject 2 R or 3 S strains into mice, which would cause illness and eventually death? Right, the virulent 3 S strains. On the other hand, injecting 2 R cells or non-encapsulated avirulent strain into the mice didn't cause any illness. The mice remained healthy. Griffith knew from the work of others that only living cells could produce pneumonia in mice. That is, if heat-killed virulent bacteria are injected into the mice remain healthy. Just like avirulent 2R strains, heat-killed virulent 3S strains cannot cause illness. Now is the critical experiment. What he did was to inject the mice of live avirulent 2R cells combined with heat-killed 3S virulent cells. Since neither cell types cause death in mice, when injected alone, Griffith expected that double injection wouldn't lead to death. Shockingly, after few days, all the mice that received both types of cells were dead. Importantly, analysis of their blood tissues revealed presence of large number of 3S virulent cells. What can we infer from this? The negative control, where we injected live 2R cells, didn't cause any illness, ruled out the possibility that avirulent 2R cells simply changed or mutated in the absence of heat-killed 3S cells. Griffith inferred that the heat-killed 3S and 2R live cells somehow interacted, he concluded that heat killed 3S cells somehow converted 2R avirulent strains into virulent 3S cells. The factor for this transformation he named as transforming principle. It was hypothesized that it might be part of the capsule or a compound for its synthesis. The transformation experiment was later refined with a soluble filtrate by other scientists. But the big question remained: which molecule serves as the transforming principle? In 1944, three scientists, namely Avery-McLeod-McCarty, obtained transforming principle in a purified state. They proved that it is DNA beyond any reasonable doubt. What did they do? The large quantities of liquid culture of virulent 3S cells were centrifuged and heat killed. Following homogenization and treatments, they obtained a soluble filtrate that still retained the ability to induce the transformation of avirulent 2R cells into live 3S cells. To eliminate all probable contaminations, the product was treated with proteolytic enzymes such as chymotrypsin, trypsin, ribonucleases or RNases. This ruled out the possibility of protein or RNA in the filtrate. Even then, transforming ability was still remained. But after treating with DNA digesting enzymes, that is deoxynucleases or DNases, it completely destroyed the transforming ability of the filtrate. This led to the conclusion that active compound in the transforming principle is DNA. Avery and his colleagues recognized the genetic and biochemical implication of this work, and they observed that transformation is heritable and affect the genetic material. We will talk about another great experiment by Hershey and Chase on genetic material in an upcoming video. For more biology learning videos, please subscribe to my YouTube channel.